Welcome to everyone who's just logging on here. We're just going to give it a moment for folks to get connected. All right, well, hello everyone and welcome to today's SSR webinar. If you have any questions about this webinar, please feel free to reach out to us directly by emailing SSR headquarters at membership@ssr.org or chatting the webinar host. This webinar is scheduled for 60 minutes and it is being recorded. It will be posted on the SSR website shortly thereafter for members to access. In just a moment, our moderator will begin with a brief introduction, followed by our speaker presentation, which will be followed by a second speaker introduction and presentation. Please feel free to submit your questions during the presentation using the Q&A text box in the bottom middle of your screen, and our moderator will read them out to the speakers when the question portions of the webinar begin. With that, I will turn things over to our moderator for today.
Hello and welcome everyone. Thank you, Erica, for that background information. So welcome to our final installation of our Rising Stars webinar series. My name is Jenna Moldvan. I'm a graduate student at Michigan State University and member of the SSR Virtual Education Committee. And it's my pleasure to serve as moderator for this session today. As Erica said, please put any questions that pop up into the Q&A and we will go through those at the end of each talk. So we're going to begin with our first speaker, which is Dr. Xiaoqi Churchill Wang. Dr. Wang received his bachelor's in animal science and first PhD in nutrition from China Agricultural University in Beijing. Following this, he moved to Texas A&M for a second PhD under the guidance of Dr. Fuller Baser and Dr. Guoyao Wu, where he studied histotroph effects on conceptus development and also worked on pregnancy recognition signaling. He then joined the laboratory of Dr. Franco de Mayo at NIEHS, where he continued to investigate uterine biology with a focus on intercompartmental signaling. Dr. Wang is currently an assistant professor in physiology of reproduction in the Department of Animal Science at North Carolina State University, where he focuses on normal and diseased uterine physiology. Today, Dr. Wang will highlight some of his current work entitled Decoding Molecular Mechanisms Controlling Uterine Aging. Welcome, Churchill.
Thank you, Jenna, for a nice introduction, and thank you for the committee to give me this opportunity to share some of our work in the lab. So today's talk will be mainly focused on mouse model. As we know that advanced maternal age, specifically beyond 35 years old for first and subsequent pregnancies, is considered a major risk factor for birth defects. And these defects included but not limited to miscarriage, late fetal and prenatal death, stillbirths, preterm births, IUGR, and even complications from maternal side, such as preeclampsia, gestational diabetes, and maternal mortality. In women over 40 years of age, actually, the incidence of spontaneous abortion can increase to more than 30%. So nowadays, worldwide, women trying to delay their first pregnancy due to a variety of factors, such as higher educational attainment, effective contraception method, increased career option, changes in values and family structure, as well as the financial and economic concerns. On top of that, lifestyle factors such as tobacco smoke and alcohol usage, as well as environmental toxins, also facilitate this process of reproductive aging. Therefore, it is imperative to understand the precise mechanism by which the female reproductive organs age, and this is a prerequisite to ultimately develop counteracting measures. So,
Churchill,
yes?
We're having some trouble with participants seeing your slides. They're only able to see a black screen at the moment.
Huh, okay.
That's weird because I'm able to see them.
Can you see now?
As panelists, we can see, but if those attendees, okay, Shuo said yes, Barbara, yes. Okay, so they can see now.
Okay, apologies for that. So for people who study reproductive aging, most of us are focusing on ovarian aging or oocyte quality, which makes sense. This is because the occurrences of embryo aneuploidy in women over 30 years and above partially can explain these poor pregnancy outcomes. However, many many evidence and research done recent years have shown that the uterine environment can also play a very important role in pregnancy outcomes with the women with advanced age. One of the fundamental study in the field is actually done by Marion Hamburger's group. And in that study, they actually found that aged mother using mice tend to have a more abnormal fetus developed during gestation. And once they actually transfer this aged embryo into the younger surrogate, those abnormal developed fetus process has been rescued. So this is a very good evidence showing that probably decidualization and the placentation defects are the major cause of age-related reproductive decline, despite of the ovarian age and over quality, which is also important. So when I started my own lab at NC State, one of the experiments we want to do is actually to see, independent from ovarian aging, during natural aging process in mice, what is the response or defects in terms of endometrium stroma cell digitalization. So we actually overexpressed these naturally aged virgin females, which are mimicking actually the first childhood for maternal pregnancy. And then we let them rest for 2 weeks, and then we give them well-controlled cocktail of estrogen and progesterone for several days to mimic this hormonal profile during normal early pregnancies. And what we see is actually independent from ovarian aging. We can see in the condition that both aged and young mice receive the same amount of steroids hormone cocktail, the aged mice could not respond well in terms of decidualization ability. You can see a small, lighter weight of decidualization in these stimulated hormones. And we actually use oil injection to physically stimulate or mimicking the blastocyst implantation process. And when we did the histology analysis, we found also they have abnormal features of the histology where you can see they have a nice decidualization occurred in these young mice, and you can see the infiltration of the monocytes for angiogenesis, where you do not see such effect in aged virgin mice. We also perform qPCR analysis to detect the progesterone receptor, which is the important molecules for pregnancies. You can see at day DD2, which stands for decidualization day 2 days after our physical decidualization stimulus, you can see progesterone receptor are downregulated, are not stimulated by the hormone treatment. So the next question is, what genes are responsible for uterine reproductive aging, or in other words, necessary to prevent premature reproductive aging? So one of the first genes come to our attention is the SIRTUN1 gene as the candidate. Why we choose SIRTUN1? So SIRTUN1 is the NAD-dependent histone acetylase acting epigenetic and non-epigenetic targets such as FOXO1, DNMT3, histone proteins, etc. And it is, even though SIRT1 does not have a DNA binding domain, it acts as a co-regulator for transcription binding to specific chromosome regions. And a previous study also showed it's involved with cell cycle, DNA repairing, insulin sensitivity, oxidative stress, as well as cell viability, senescence, and inflammation during aging. Study using yeast, flies, and mice also show that SIRT1 can delay cell senescence and extend lifespan of such subjects. An in vivo study in humans suggests that SIRT1 can interact with estrogen receptor and progesterone receptor to affect their respective transcription activity. So in null mice study, the female SIRT1 null mice are subfertile. However, the uterine specific role other than the ovarian function of the SIRT1 during pregnancy is still unknown, as well as its perspective on uterine aging. So first, the thing we want to test is whether SIRT1 expression level was decreased in our aged uterine deciduoma. And you can see on the left, the aged, this is done by RNA-scope, showing the mRNA location of the SIRT1. You can see SIRT1 expression is downregulated in the aged uterine deciduoma, and we validated this using also qPCR analysis. So the second question is, what's the pattern or temporal spatial expression pattern of the SIRT1 during normal pregnancy in the young mice. We actually test that using the young mice, and then you can see that the SIRT1 actually expressed throughout the peri-implantation period of pregnancy in the young mice, and they are located at epithelial stroma cell, luminoepithelial, and glandular epithelial cells. You can also see that SIRT1 is highly expressed in the embryo. So now we form this hypothesis that SIRT1 could play an important role in governing age-related uterine receptivity, and the uterine-specific ablation of SIRT1 could accelerate this premature uterine aging. To test this hypothesis, we actually generate a conditional knockout mice for SIRT1. We're using progesterone receptor Cre to knock out the SIRT1 specifically in the uterus, in the progesterone receptor positive cells in the uterus. And you can see our knockout is pretty clean. And once we actually generate mice, we put them on a 6-month breeding trial. And you can see during this 6-month breeding trial, the female mice lacking uterine SIRT1 are producing less liters per female, less pups per liters, and increase the stillborn pups within the 6-month breeding trial. Once we actually look further into the data of the 6-month breeding trial, we can see that SIRT1 deficient females actually have this defects in liter frequency and pups per liter at specific liters, and also accumulative live pups born per female. And we also found 6 out of our 7 SIRT1 knockout mice become sterile after giving birth to their 3rd litter, and the timing actually around 25 weeks of age, which is the prime time of their reproductive life in mice. So further, we wanted to investigate what's the cause of this phenotype of this subfertility. First thing we did is actually to see whether there's impact of SIRT1 on embryo implantation and anything related with spacing. So seems like the embryo can implant at least the number of implantation site was not changed between SIRT1 knockout and the wild type. And we did see that the decidual bulb weight are much less than the control mice. And we also see this uneven spacing only occur significantly at day 9.5, since the spacing is actually, the embryo spacing is actually fixed during implantation. These changes may due to this embryo absorption during the decidualization process. So we further look into the day 5.5 and 6.5, which is after a day or two after implantation of the blastocyst, and we can see that SIRT1 actually impact the embryo in vision. And later at 9.5, where the placenta is actually start to differentiate and form, and we can see this placenta is abnormal and they are off the center. At least the trophoblast portion is much smaller than the control mice. And we think this might be due to the defects in decidualization process, because the embryo is not deficient from SIRT1 deficiency, SIRT1 expression. So to test that, we actually performed the artificial decidualization assay. You can see on the right that they did actually, by knocking out SIRT1 in the uterus, it did actually have a defect in decidualization. And so we know that aging is a gradual loss of molecular fidelity, which eventually causes the disease and death of the individual. So this is not a black and white or yes or no question when we investigate the molecule changes. So we look into 4.5 at this time. We actually suspect whether this decidualization defect is happened earlier or at the time of implantation occur. So PTGS2 is the first molecule investigated because it's the marker for implantation. And you can see on the left, in a normal situation, the PTGS2, which is another name COX-2, the enzyme for prostaglandins. So you can see the PTGS2 at this time is move away from the luminoepithelial cells into the stroma cell right beneath it. And you can see this single columnar luminoepithelial is actually under morphological changes into this ready to undergo apoptosis for invading embryos. Whereas you can see the SIRT1 knockout mice, they actually remain the intact structure of single columnar lobed epithelial cells, and PTGS2 is still remain in the epithelial cells. So this is a clear sign for implantation delay. So another pathway we are interested in is the FOXO1 and PGR expression. So previous study have shown that there is a yin and yang expression patterns between FOXO1 and the progesterone receptor, which is required for successful implantation of the blastocyst at 4.5. So normally at 4.5 pregnancy, FOXO1 actually will shuttle from cytoplasm into the nuclei at the epithelial cells. And in return, the progesterone receptor actually will disappear from the same location. This is what we expect in normal condition. So once we knock out the SIRT1 in the uterus, what we see is that this yin and yang balance has now been broken. And you can see the FOXO1 now is not high enough in the nuclei, and they become very patchy in the luminal epithelial cells, whereas progesterone receptor actually back to or come back in the same place. So this is important. This interference of the imbalance of the FOXO1, yin and yang balance of FOXO1 and progesterone receptor is critical for successful implantation. So now we think, okay, so they have defects in implantation. However, they do implant, and whether these defects or suboptimal condition was caused earlier than the time of implantation. So we next move to the window of receptivity. So in the case of mice, it's day 3.5 of pregnancy. So window of receptivity defined as the period that's progesterone acting on progesterone receptor on luminal epithelial cells and stroma cells. So luminal epithelial cells will now stop proliferating, become permissive for blastocyst attachment and invasion. At the same time, it will prime the stroma cells for dehydrogenation. Many labs have actually studied these complicated molecular pathways. So we actually propose that SIRT1 could, in fact, affecting proliferation as well as dehydrogenation during this window of receptivity. We did found that SIRT1 actually interfere with estrogen and progesterone signaling pathway, in which they will increase the activity of estrogen as well as downregulate progesterone signaling. At the same time, we actually investigate the Indian hedgehog signaling pathway by looking at the PATH1, which is downstream receptor for Indian hedgehog, and as well as COOP-TF2, which is a key molecules that's required to express in the stroma cells for successful priming the stroma cell for dehydrogenation. However, we do not see very clear patterns for FGF factors expression. Normally, they will be inhibited to actually remove the stimulus for proliferation in the epithelial cells. So instead of looking those patterns, we're actually directly looking to the proliferative index by looking at the Ki67. What we see is the sporadic expression of this Ki67 once we knock out the SIRT1 in the uterus. And we also quantified it. We found stroma cell had less expressed of Ki67 in terms of deficient SIRT1 expression. So this makes sense because we do see the knockout of SIRT1 mice have pups. So meaning the embryo can implant, but just have suboptimal conditions for that. So now we know the Indian hedgehog pathway has been affected. What about other molecules or other pathways that associate with window of receptivity and later affecting embryo invasion, spacing, and placentation? So we performed the bulk RNA sequencing, and we found that estrogen signaling actually was upregulated by SIRT1 knockout, and progesterone signaling also downregulated by SIRT1 deficiency. We're also validated, our results also validated Indian hedgehog signaling has been compromised, as well as adrenomagnetin signaling, hyaluronic synthesis, and epithelial cell junctions. So when we perform this top enriched functional annotations, we found that by knocking out or deficiency in SIRT1 in the uterus, we found activation of the somnolation pathway, autophagy, senescence pathway, and then we also found, of course, SIRT1 pathway has been inactivated, notch signaling, hedgehog, as well as regulation of epithelial mesenchymal transition that's ready for embryo to implant. So we actually compare our current transcriptome data with the other public available transcriptome data in uterus at day 3.5. We found aging is actually ranking #1 with the top overlapping numbers of transcriptomes with our uterine transcriptomes SIRT1 knockout. And this is actually demonstrated that SIRT1 actually a SIRT1 knockout in the uterus deficiency is actually mimicking or partially mimicking uterine reproductive aging. So we next perform the pixel series red staining to actually show that there is an accelerated deposition of age-related fibrillar type 1 and type 3 collagen in the SIRT1 deficient uterine stroma. So I hope I convinced you that SIRT1 now actually deficiency of SIRT1 actually mimicking reproductive aging in the uterus. Recently, we also performed the transcriptome analysis with our naturally aged mice, but it's actually under the fine treatment or control of hormone cocktail. So we performed the ovarectomy first to remove the factor for ovarian aging, and then we give them the shots of estrogen and progesterone to mimic day 3.5 of pregnancy. What we found is actually among those upstream regulators, SIRT1 is ranked very high, actually ranked #2 for upstream regulators that actually control this control the reproductive aging. And for the hyperactivated pathways, there are androgen receptors, and there are pro-inflammatory factors and anti-inflammatory factors, as well as the p53. And then we will look at the bubble chart of a canonical pathways that are affected by aging. What we can see is a lot of pathways that's being activated are involved with immune response and cancer, as well as the immune system. And for the pathway that has been suppressed or inhibited, that's mainly focused or located in the cell cycle. So in conclusion, the age-related dysregulation defects are correlated with downregulation of SIRT1. By deleting the uterine SIRT1, we believe we generate a genetic aging mouse model that can recapitulate physiological aging of the uterus, leading to impaired implantation and stroma cell dysregulation. So in future study, we wanted to know specific roles of SIRT1 in the epithelial as well as in the stroma cells, and how SIRT1 interact with factors in progesterone receptor signaling pathways such as acetylone, epigenetic modification, etc. We also want to know SIRT1 responsive cis regulatory elements as well as its transcriptional cofactors. Eventually, we want to identify gene signatures sensitive to reproductive aging, and then eventually develop the counteracting measures. So with that, I'd like to thank my lab members. The majority of the work has been done by my student, Maggie Cummings, as well as other lab members. I also like to thank my collaborators for their support, and of course, our funding agency for financial support. Thank you very much.
Great talk, Churchill. Thank you. While we wait for some questions to roll in, I'll start off with one here. So SIRT1 in humans is associated with acetylation of NF-kappa B, and that prevents chronic inflammation and stress resistance. So do you think that there's a potential that SIRT1 in the murine uterus is perhaps regulating innate immune response, and that could be affecting maternal remodeling associated with deciddualization, implantation, and possibly post-parturition repair?
Yes, that's a good question. Actually, from our human data, we didn't show, as well as our downstream analysis, NF-kappa B is one of the target that by SIRT1. So it does, well, SIRT1 is a deacetylase, so they will actually modify tons of molecules. And I believe NF-kappa B is a critical one that has been remodified by SIRT1.
Okay, there's no other questions. I'll ask another one. What do you think are the compartment-specific roles of SIRT1? I know you said you're planning to investigate that, but within the murine uterus, do you think that SIRT1 is playing alternative roles in either the stroma or the epithelium?
Well, from our progesterone receptor PR3 knockout, we suspect it's more in the stroma cells. However, whether this stromal cell or dysregulation defect is from stroma cell itself or from the miscommunication between epithelial cells and the stroma cells is still in question. We actually have the lactoferrin-3 knockout SIRT1, which achieved the similar subfertility. And we now actually in the process of actually generate the stroma knockout only to actually see whether these defects is due to the communication between epithelial cells and stroma cells or solely due to the stroma cells. And we also have human endometrial stroma cell experiment actually to back up our hypothesis. So I think in short answer, yes, stroma cell dysregulation is a major effect by SIRT1 other than implantation, but these could be due to both communication between two compartments and also solely depend on stroma cells during dehydration.
Okay, great. Thank you so much, Churchill. We're going to move on.
Thank you.
Oh, sorry, we've got one question that came up, so let's do this one real quick. Thiago asked, does SIRT1 have a role in the uterus of animals attaining puberty?
That's a good question because PR is actually expressed the post-neonatal stage. We do not see morphologically there are no difference affecting the uterus, but I don't think so. Yeah.
Okay. All right. With that, then we'll conclude. Thanks again, Churchill. And we'll move on to our next speaker. Okay, great. Okay, so our next rising star today is Dr. Amanda Patterson. Dr. Patterson completed her bachelor's in equine sciences and master's degree in animal sciences at Colorado State University. Dr. Patterson then moved to Washington State University to complete her PhD under the guidance of Jim Prue, where she studied endometrial regeneration. Following the completion of her PhD, Amanda moved to Michigan State University as a postdoc in the laboratory of Dr. Jose Teixeira, where she studied stem progenitor dysregulation in the development of uterine fibroids. Dr. Patterson is currently an assistant professor in the Division of Animal Sciences and Department of Obstetrics, Gynecology, and Women's Health at the University of Missouri, where her research focuses on endometrial regeneration. Dr. Patterson's talk today is entitled Towards Understanding Mesenchymal Epithelial Transition and Endometrial Epithelial Regeneration. Welcome, Dr. Patterson.
Thank you, Jenna. Good afternoon, everyone. First, I'd like to thank the organizers for the opportunity to share my research, and then also to Jenna for moderating today. So I don't think I need to go into great detail with the group here, so I just want to briefly introduce endometrial regeneration and why we're studying it. So very generally, endometrial regeneration is the restoration of the functional layer of the endometrium following desquamation, as we can see depicted in this cartoon here during the menstrual cycle. We are focusing on regeneration of the epithelium, termed epithelialization in particular. Epithelialization during endometrial regeneration occurs during menstruation in women and following parturition in most eutherian species. And our overarching goal is to understand the mechanisms involved in endometrial regeneration so we can gain insight into disease pathogenesis when these mechanisms become dysregulated. Briefly, there are several proposed mechanisms of epithelial regeneration. The first being replication of glandular and luminal epithelial cells that escaped desquamation. The second is contribution of endogenous epithelial stem or progenitor cells. The third is mesenchymal to epithelial transition, which is what I'll talk about today. And lastly, there's possibility of contribution by exogenous bone marrow-derived cells. And it's likely that multiple mechanisms are at play to quickly and efficiently repair the epithelium, in particular the luminal epithelium during regeneration. So mesenchymal to epithelial transition, or MET, and the reverse process, which is EMT, or epithelial to mesenchymal transition, are critical for embryo development as well as tissue and organ formation, and are also important in wound healing in the adult. Interestingly, they're also exploited by diseases such as cancer and fibrosis. So EMT and MET are types of cellular transdifferentiation whereby one cell type will transition into a completely different cell type. And this cartoon here depicts the basic cellular changes associated with transition. On the left, we have epithelial cells, and on the right, we have mesenchymal cells. And I'll describe the EMT process as it's more well studied. So an epithelial cell is characterized by having cell-cell adhesions, predominantly through these tight junctions and adherence junctions. They have well-defined apical basal polarity. They sit on a basement membrane and they're stationary. As the epithelial cells begin to transition into mesenchymal cells, they'll lose their epithelial characteristics while gaining mesenchymal characteristics, being loss of cell-cell adhesion, loss of the apical basal polarity, and they'll become spindle-shaped. They detach from the basement membrane and are surrounded by extracellular matrix, and they're migratory. And then, of course, the reverse process is MET, mesenchymal cells becoming epithelial cells. So I'm going to start off with sharing the seminal studies of MET in endometrial epithelial regeneration that were conducted during my PhD studies in Dr. Jim Prue's lab at Washington State University, and also studies at the same time from Richard Behringer's lab at MD Anderson. So what we did in Dr. Prue's lab is we used the AMHR2 Cree ROSA 26 flock stop EYFP reporter system. AMHR2 is active in the mesenchymal cells of the uterus, so that would be the stroma and the myometrium. And upon Cree activation, we'll get YFP expression in mesenchymal cells and mesenchymal-derived cells. And we can see in peripubertal mice here that the stroma is abundantly expressing YFP. The myometrium, not seen very well, also expresses YFP. And you can see that the epithelium is devoid of YFP. Richard Behringer's group used a similar mouse model, AMHR2 Cree mice, with a LAC-Z report, and showed that in nulliparous mice, similar to ours, there was abundant LAC-Z expression in the mesenchyme, but no expression in the epithelium. And then both of our labs showed that after pregnancy, in which there is endometrial regeneration that occurs, we identified labeled epithelial cells in a mosaic pattern of expression, both in the luminal and the glandular epithelium. And so this suggested to us that the labeled stromal cells had undergone MET to become epithelial cells during endometrial regeneration, resulting in the labeled YFP positive epithelial cells. In the same study, we used CD1 mice, and we used a Menzies-like mouse model where we induced pseudopregnancy, induced decidualization, and then allowed the decidual tissue to grow, and then OVEX the mice, removing progesterone stimulus that allows the decidual tissue to degenerate and the endometrial tissue to regenerate. And what we identified were transitional cells in this regeneration process. So this is 48 hours into regeneration. This white line is demarcating where the regenerating epithelium where the regeneration of the epithelium will occur, and then where the deciduoma is located. And we identified keratin positive cells located in the stroma. In the middle is showing vimentin, and then we can see higher magnification images showing that these cells co-express cytokeratin and vimentin, suggesting that they're in transition from mesenchymal to epithelial cells. And these cells increased across the time of regeneration out to 48 hours, and regeneration is typically complete by 72 hours post-OVEX. So now I'm just going to summarize what we've learned over the past 12 years studying mesenchymal to epithelial transition. So these first 2 points I just discussed from those seminal studies, as well as Philippa Saunders' lab also used the Menzies model to identify vimentin and keratin positive transitional cells. And in that study, they also showed expression of MET and MET markers during regeneration. A paper out of Yale suggested that stromal or mesenchymal stem progenitor cells may be the cells that give rise to those that undergo MET to become epithelial cells. Philippa Saunders' lab then recently published using a refined lineage tracing mouse model that I'll discuss in the next slide, and also used single-cell RNA-seq to investigate MET. And then lastly, we recently showed that MET may also contribute to epithelialization in the estrous cycle, where there's epithelial turnover due to apoptosis and proliferation, but no endometrial shedding, and it may also be involved in adenogenesis. So our overarching question moving forward beyond these studies is, are epithelial cells that are derived from the mesenchyme, are they bona fide endometrial epithelial cells? So to begin addressing this question, we decided to transition from the AMHR2 Cree reporter mouse model because of some important caveats. An important one is that AMHR2 Cree has embryonic activation. And as I just described, we published that MET may contribute to epithelialization in the estrous cycle and adenogenesis. And so using this mouse model, we're not able to distinguish between de novo MET during endometrial regeneration and proliferation of mesenchyme-derived epithelial cells that originated during earlier MET events, such as during adenogenesis or the estrous cycle. So we decided to use the PDGFR alpha Cree ERT2 tamoxifen inducible system with TD tomato reporter. So for those not familiar with this system, PDGFR alpha is a mesenchyme specific promoter. It drives expression of a Cree estrogen receptor fusion protein. The estrogen receptor is mutated so that it responds to tamoxifen. So without tamoxifen, Cree does not enter the nucleus, but upon tamoxifen treatment, it will enter the nucleus, cleave out the stop sequence, and then we get tomato red expression in mesenchyme and mesenchyme-derived cells. And as I mentioned, Philippa Saunders recently published using this mouse model in the menstruation-like mouse model, along with single-cell RNA sequencing. So back to our question, are mesenchyme-derived cells, or MD epithelial cells, bona fide endometrial epithelial cells? So we're using this PDGFR alpha Cree ERT2 reporter mouse line, which will allow us to track the transition of labeled mesenchymal cells into epithelial cells that will retain that label specifically during endometrial regeneration. We use a pregnancy mouse model so that we can assess the MD epithelial cells after regeneration is complete, which is not possible with the OVEX Menzies model because there's no longer hormonal stimulation of the uterus. And then we also use single nuclei RNA-seq so that we can investigate the mechanism of MET and assess the similarities and differences between the MD and the non-MD derived and the non-MD epithelial cells in the reporter mice, because we can use the TD tomato gene expression to identify our cells of interest. So for the next bit of this talk, I'm just going to cover what we've done so far using this new mouse model. This work is spearheaded by my postdoc, Sadao Wang, and we started off by characterizing the MD or mesenchyme-derived epithelial cells during postpartum regeneration. So for our timeline, we took sexually mature mice, treated them with tamoxifen for 3 days. They then underwent a 4-week washout to remove tamoxifen effects on the uterus. They were then either collected during the cycle or were mated to males of proven fertility. And then those mice were collected on E18.5, which would serve as our time 0 for regeneration. And then also collected on postpartum days 1, 2, 3, and 4 during regeneration. And by day 4, regeneration is complete. And then we collected day 28, which is after regeneration is complete and the mice have undergone a few estrus cycles. And what I'm showing you here are flow cytometry scatter plots of our gating strategy. So we identified our live cells, our EpCAM positive epithelial cells, and then tomato red positive cells, which would be our stromal or mesenchyme derived cells. And we looked at the presence of the MD epithelial cells, so EpCAM positive, tomato red positive cells during our different time points. And what we found is that there are low levels of MD epithelial cells present at E18.5, but there's a significant increase during postpartum day 1 of regeneration with a peak at postpartum day 2. And then they fairly rapidly decline such that by the time you get out to a normal cycling mouse, there's significantly fewer MD-derived or MD epithelial cells. And so this suggested to us that MD epithelial cells arise very quickly during postpartum regeneration, but are transient, suggesting that they're not functional endometrial epithelial cells. So we asked the question, does this increase in MD epithelial cells early during postpartum regeneration, is that due to this presence of cells at E18.5 proliferating, or do we actually have MET occurring? So we isolated cells on postpartum day 1 and sorted them into the tomato red positive MD epithelial cells and the tomato red negative non-MD epithelial cells, and quantified organoid number and organoid size at day 7 and 14 of culture. And we plated these such that these should be organoids that derived from individual cells. So we can see that there are significantly more organoids that are formed in the non-MD epithelial cells compared to the MD epithelial cells. And similarly, the organoids that form are significantly larger from the non-MD epithelial cells compared to the MD epithelial cells. And we also looked at proliferation during postpartum day 1 in the tissue sections. And this was done by Ki-67 staining. And similar to our organoid study, we can see that the non-MD epithelial cells proliferate significantly more than the MD epithelial cells. So the low proliferative index of the MD epithelial cells, the mesenchyme-derived epithelial cells, suggests that the increase in these cells during postpartum regeneration is due to continued MET, not due to proliferation of previously established MD epithelial cells. So moving forward, we performed single nuclei RNA sequencing of the tissues during postpartum regeneration, with our goal being to investigate the actual process of MET. Again, this is using the tamoxifen-inducible reporter mice. And on the right, I'm showing a UMAP compilation of cells from embryonic day 18.5 and postpartum days 1 through 4. We can see several of our classic cell types that we would expect to see in the uterus, mesenchymal cells, endothelial cells, various immune cell populations, epithelial cells, including glandular epithelial cells. And then importantly, we found what we believe to be a transitional cell population. So investigating that population a little more, we looked at expression of different epithelial cell markers. On the top, we're seeing 2 pan-epithelial cell markers, being keratin 8 and keratin 7 expressed by these cells. And interestingly, they do not express more of the classical stationary epithelial cell markers, being CDH1 and EpCAM. And I'll come back to these 2 markers in a few slides and discuss them a little more. In regards to mesenchymal markers, we can see vimentin expression as well as collagen 1A2. And then when we look at a series of mesenchymal and epithelial cell markers, we can see that these transitional cells are still predominantly mesenchymal in phenotype, which will make sense when I show you the next slide, where we appear to see them migrating. Remember, mesenchymal cells are migratory, but they do start to express low levels of various epithelial markers. And importantly, these cells express tomato red, indicating that they originated from the labeled mesenchyme cells in our reporter mice. Other markers of interest that they expressed are stem cell markers, so ALDH1A1, TERT, and CD34. And really interesting to us was the expression of Wilms tumor 1, which is well known for its role in various cancers. But recently, it was shown to drive progenitor cell progression and epithelialization in kidney organoids. And so we believe that our preliminary data is showing the presence of a transitional population containing differentiating progenitor cells that may have originated from a mesenchymal stem progenitor cell. So going back to our tissue sections, we wanted to see if we could identify these transitional cells in the tissue. And what I'm showing you here is an example of one mouse. This is postpartum day 1. And we can see keratin 8 expression nicely highlighting the luminal epithelium. And interestingly, we found these keratin 8 positive cells in the stroma, very similar to what we saw in our Menzies model using the AMHR2 Cree mice. And what was really interesting is that they appear to be in this migratory fashion towards the epithelium that is regenerating, potentially contributing to that regeneration. Now, not all of our tomato red is very highly expressed, and so we used RFP antibody to enhance the tomato red signal so we could confirm that these cells express tomato red, indicating that they are derived from our labeled mesenchyme or stromal cells. So again, cytokeratin 8 in green, and then RFP in magenta, and we can see at high magnification that many of these cells also express RFP, indicating that they were derived from tomato red expressing mesenchymal cells. Looking at these cells in a little more detail, not all of this am I able to show here, but we looked for CD45 expression, showing that they're not immune cells, cleaved caspase 3, showing that they're not undergoing apoptosis. I showed you the tomato red expression. They also, a good number of them express vimentin, and most of them are spindle shaped. So this aligns nicely with our seq data, suggesting that they're still quite mesenchymal as they're migrating to contribute to the epithelium. And then, of course, they express keratin 8. That's how we identified them. And they do not express EpCAM or CDH1. And these are markers of stationary, fully transitioned epithelial cells. And this aligns with our seq data. Now, what's really interesting in this figure here, where we're seeing keratin 8 and CDH1 expression, we can see the transitional cells outlined in white, expressing keratin 8, not expressing CDH1, which is E-cadherin. However, I'll draw your attention to this little box here, where we can see a cell that expresses keratin 8, again, not expressing CDH1, but a cell directly next to it that appears to be incorporating into the luminal epithelium, expressing keratin 8 and also expressing CDH1. And so these data suggest that the keratin 8 positive cells in the stroma are cells in partial MET, still retaining more mesenchymal characteristics, but then upon reaching the epithelium, they complete the transition and will upregulate the more traditional epithelial stationary cell characteristics. And so to conclude, what I hope I've been able to show you today is that the mesenchyme-derived epithelial cells arise de novo through MET, not proliferation during endometrial regeneration. And this suggests that MET is a process of terminal differentiation resulting in MD epithelial cells with low proliferative potential. And that goes in line with the second point that these MD epithelial cells are transient, suggesting that MET serves as a mechanism to help quickly repair the epithelium to reestablish that luminal barrier, which is important for preventing infection, but that these MD epithelial cells are quickly replaced and therefore don't likely function as classic endometrial epithelial cells. And then lastly, we identified transitional cells in the process or using sequencing, and these cells expressed pan-epithelial markers, but not stationary epithelial markers, suggesting that they're migrating to the epithelial layer. Then upon reaching the epithelial layer, they appear to express the stationary epithelial cell markers as they incorporate into the epithelial layer. And lastly, they express stem progenitor cell markers, suggesting that a mesenchymal progenitor cell may be the source of the cells that undergo MET. And with that, I will just thank my lab. As I said, most of the work was conducted by Zadao Wang. I also have a master's student, Mackenzie, who is currently taking up a human side of this project to assess MET using human cells. And of course, my collaborators at MU, Oklahoma State University, and LSU, and then our funding source. And I would be happy to take any questions.
Thank you, Amanda. That was a great talk. While everyone has a moment to think about some questions, I'll ask one to start. So your evidence suggests that these MD epithelial cells are coming from a stem or progenitor population, as you said. Do you think that this is a specific stem niche that's found in either the myometrium or the endometrium? Where do you think these cells are coming from?
That's a good question. So the hypothesis is that mesenchymal stem progenitor cells in the uterus are located in the perivascular niche. And so that would be my go-to thought that these cells could be arising from those perivascular cells. What I will say though, so there was a paper by Pradeep Tanwar where he used CSPG4 mice, which mark perivascular cells. Now he didn't do any regeneration in those studies, so he didn't see any MD epithelial cells. But it would be interesting to follow up with those mice and see if under a regeneration event, if they contributed to the epithelium. But I actually think that it's probably less of a stem cell and more of a progenitor cell that's maybe more differentiated and could have a more varied niche environment. But with our mice, we can't really narrow that down yet at this point in time.
Very interesting. Thank you. I'm going to give it another moment to see if any lingering questions appear for Amanda. Okay. Barbara said, I think you mentioned these cells are transitionary. Do the permanently labeled cells accumulate over time in the epithelial layer in older mice?
That's a great question. Based off of looking at least 28 days after regeneration has occurred, and the population has substantially decreased, we don't think that they're retained long-term in the epithelium and likely don't increase over time. But that's something that we're following up with to see if they do happen to fluctuate perhaps across the estrus cycle, similar to what we see with our other mouse model.
Okay, we have another question from NIEHS from Humphrey's lab. Ewing Chen asked, do you see more MET around implantation sites? And if so, does the number of tomato cells correlate with litter size?
That is a great question. Using our previous mouse model, which again has issues with it, we did not see any differences around implantation sites compared to inter-implantation sites, which we actually hypothesized that we would see more MD epithelial cells at the implantation sites because there would be more damage and repair. We are currently following up that study with this mouse model to determine if similarly there aren't differences. But considering that there is some turnover of the whole epithelium during pregnancy, it's likely that we could have MD cells, MD epithelial cells present in inter-implantation sites as well.
Okay, and now Humphrey has asked a question. Did you observe any regional specific contribution of these mesenchymal cells, as in mesometrial versus anti-mesometrial?
So typically we see A little more in the mesometrial pole, but I couldn't say that it's specific enough that we could actually say it happens more in the mesometrial pole. So a little bit more in that area, but it does happen in both mesometrial and anti-mesometrial pole. We see MD epithelial cells in the glands as well, which those residing more in the anti-mesometrial pole gives us evidence to suggest that it's happening down there as well, but more so in the mesometrial.
Okay, it looks like we have sufficiently answered all the questions. Thank you guys both for taking the time to share your research today, and congratulations on being one of SSR's Rising Stars. And I'll turn it over to Erica to close us out.
Awesome, wonderful. Thank you all so much for joining us, and that will conclude our webinar for today.
Thank you.